In this part of the lecture, we're going to look at how an action potential stimulates a muscle to contract, and then we're going to look at what's called the sliding filament theory, about how muscle contraction actually happens. So let's come back to this. Well, let's go back to where we were before, just so we know where we're going. This is the slide we ended on in the last section, and here we have a neuron with its axons coming in and associating with this muscle fiber. And we mentioned that this axon terminal does not quite touch the sarcolemma. It comes really close to the muscle fiber, but they don't actually come in contact. So we have this little space. That space is called a synaptic cleft. So in the first part here, we're going to look at what happens at that synaptic cleft. So let's move to this slide here. Let's get our bearings first. So this is the axon terminal coming around here. This whole part here is our muscle fiber. So we have our axon terminal, or our neuron, coming in and getting really close to touching the sarcolemma. So this very upper edge here, that's going to be the sarcolemma. But we have this little gap, and we call this gap the synaptic cleft. So they never do actually come in contact with each other. What this means is we have a electrical signal coming down from our brain through our neuron to tell this muscle to contract. But because we have this little space, that electrical signal isn't what actually stimulates the muscle. So once that electrical signal gets to the axon terminal, we have to change that signal, because we have to send something across this little space in order to activate the muscle. So what happens is this nervous signal, this electrical signal of the nervous system here in the axon terminal, is turned into a chemical signal. And we have something called a neurotransmitter. And this neurotransmitter is what's going to be is a small chemical which goes across the synaptic cleft from the axon terminal to the muscle fiber. So what this nervous system signal actually does is it signals the release of this neurotransmitter. So our brain says, okay, let's contract a muscle, sends a signal down. When it gets to the axon terminal, that stimulates the release of these little packages. So these are little vesicles filled with neurotransmitter, and they're going to move to the plasma membrane of the axon terminal, and as they fuse with that membrane, that's going to release their contents, the neurotransmitter, into the synaptic cleft. And then the neurotransmitter is going to move across the synaptic cleft, bind with some receptors on the sarcolemma, and then that's going to stimulate the muscle to contract. Also notice these big tubes called T-tubules, and they go down inside, so they're openings on the outside of the muscle fiber that move down deep inside the muscle fiber. And they have an important job, because as this signal is moving across, once the neurotransmitter signals this muscle and it starts sending a signal along the muscle, this signal is going to go down the T-tubules deep inside the muscle fiber. So we have an electrical signal coming here, changing to a chemical signal, moving across the synaptic cleft, and that's stimulating the muscles, and the muscles also send an electrical signal. That's how the muscle is stimulated. So we go electrical, chemical, and then we have another electrical signal moving down the muscle fiber, and that's causing the muscle to contract. Let's look at a little bit more about the neurotransmitter. So this is this is a chemical that is released from the axon. This chemical is released into the synaptic cleft, and there it eventually goes and binds with the sarcolemma. So in this picture here, this area here is our muscle fiber. This is our muscle cell. We have the sarcolemma up here at the top. This is our plasma membrane of the muscle cell. Up here we have the plasma membrane of the neuron. And this up here is going to be your axon terminal, as you can see pointed out here. And then here's our synaptic cleft, this little tiny space between. So our signal came from the neuron, from the brain, says, okay, release this neurotransmitter. This neurotransmitter comes out into the synaptic cleft. On the sarcolemma, we have these receptors. And this neurotransmitter, this chemical, is going to bind to that receptor. And that's what triggers the muscle and says, so when you get these little chemicals on those little on those receptors, that triggers the muscle cell to send its own electrical signal along the muscle, which is going to cause it to contract. This neurotransmitter is called acetylcholine. And we abbreviate this as a capital C, and an H. So acetylcholine, we'll see this again in the nervous system, so this is a good time to learn that name. So acetylcholine is a neurotransmitter in the case of muscles. So once this acetylcholine attaches, that's going to signal the muscle, and the way the muscle is signaled is by potassium and sodium, the relationship here. So acetylcholine allows potassium to rush out of a cell and sodium to rush inside a cell. These are charged ions, and so that's what causes this electrical signal. And again, we'll talk more about this in the next chapter, but I just want to point out that sodium and potassium, this is the same for the neurons, but sodium and potassium in their relationship, they're the major ions of our electrical system. One other thing to point out here is that so acetylcholine comes and attaches, but this attachment causes the signal. So how does the signal end? Well, acetylcholine gets broken down over time. So once it comes and it attaches, then it's immediately going to start being broken down, and you can see that in this area here. So there's another enzyme that's attached, a protein enzyme attached to the sarcolemma, and its job is to break the acetylcholine apart so it's no longer active. So when it attaches to this enzyme, it breaks it apart into acetic acid and choline. These are then going to be reabsorbed into the axon terminal, where they can be reproduced into more acetylcholine. So this is kind of a little merry-go-round here. So our acetylcholine comes out, it attaches, signals the muscle, comes over here, gets degraded, then it's back up here into the axon terminal, and then eventually, when the next signal comes, it can be rebuilt and sent back out again. So this process is constantly going on inside our muscles. So how does that nervous electrical signal translate over to a muscle contraction? It comes back to the actin and myosin that we already have talked a little bit about and we've seen. So the myosin heads, when they are stimulated by a signal from the nervous system and then the electrical signal moving down the muscle fiber, those little myosin heads bind to the thin filament or the actin filaments. Let's go back to this previous picture that we were looking at to remind you where the actin and the myosin are. So here we have the actin, which is this blue thin filament, and then we have the myosin, which is the red, and it has all these little projections. Those are called the myosin heads. So when that electrical impulse comes, what that does is it causes these little myosin heads to bind to the thin filament. And then they're going to start moving, and they'll actually pull these thin filaments closer together. That is how the muscle contraction happens. So the activation causes the myosin heads to attach to the thin filaments, and then they start pulling and moving those actin filaments closer together. And then this causes the actin to shorten, which causes the sarcomere to shorten. And I guess I shouldn't say it causes the actin to shorten, but it causes the actin to come closer together, which causes the sarcomere to shorten, which causes the microfibril itself to shorten, which causes the muscle cells to shorten, so that finally, eventually, the whole muscle is contracting and shortening. And this is happening all at the same time. So let's look at this here. So in this picture, we get our bearings here. So the red is the myosin, and you can see those little projections, the myosin heads. The actin is the blue here. We have the H zone, the area where it's just myosin, there's no actin. We have the A band, the dark band, where the myosin is and it's overlapped by the actin. We have the I band, the light band, and that is just where there is actin. And we also have the Z disc here as well. So what's happening here? So our electrical impulse comes, these little heads are going to attach to the actin, and they're going to start pulling the actin closer together. The actin doesn't change shape, it doesn't actually shorten, but it just starts being pulled closer together. So what happens is we end up with this here. So as they shorten, our sarcomere, which goes from here to here, that's one sarcomere. Notice our sarcomere is shorter. If you look down at this lower picture, before we were this wide, now we're shorter. Look at the A zone. The dark band actually has not changed length. The A band is still the same size. And that's because the myosin doesn't change shape, it doesn't shorten at all. So the A band, the dark band, is the same length. But then look at the I band. So originally the I band started here, and it went to around here somewhere. But look in our new one, not our new one, but in our contraction, after our contraction happens, our I band has shortened to here. Look how much shorter it got. And this is true on both sides. I band was from here, and then it came, and now it's just this small. So the I band is what gets smaller, and as that I band shortens because the actin is being pulled closer together, that causes a contraction. Notice the H zone as well is originally this wide, but now it is basically non-existent. The H zone has disappeared because the H zone is the area where there's no actin overlapping. But when this contraction happens, the actin totally overlaps and removes that H zone, so it's no longer there. So when a contraction happens, the I band is shortening, the H zone is disappearing, the A band is staying the same, and this is a contraction. So this sarcomere, that causes the shortening of the sarcomere, and this is going to happen on all the sarcomeres, the length of the muscle. And then as this happens in all the sarcomeres, this leads to the shortening of the muscle as a whole. And this is going to last as long as the stimulation lasts, but then once the signal to contract is over, then this muscle is going to go back to its previous shape. The myosin is not going to be attached to the actin anymore. It's going to release and it's going to move back. That is how muscle contraction happens. This might be a little bit complicated to think about. It's kind of hard to picture as well. It's good to watch some videos. There's a lot of videos about this sliding filament theory that you can watch. So I encourage you to go on and look at the videos and listen to this recording as many times as you need to as well to get a feel for how this sliding theory works. But the main idea here is that these proteins here, these actin and myosin, are working together to cause the shortening of this muscle. That's how the contraction happens. One other thing to mention in this sliding filament theory, and that is the role of calcium. Calcium plays an important role in this whole process here. In a resting muscle, the actin and myosin cannot bind. And the reason is because you have these little regulatory proteins here. Here they're purple, little purple proteins that are in the way. So it looks like the myosin is attached to the actin here, but the binding site is being covered up by this little protein. So at this point, at this resting stage, this is a resting stage here, at the resting stage there's no contraction happening because the actin and the myosin can't bind to each other. The myosin heads can't bind to the actin. Calcium needs to be released in order for these binding sites to be opened up. The calcium is stored in the sarcoplasmic reticulum. and it's found throughout the muscle cell. So it's the area of the cell where the calcium is all being stored, these calcium ions. When the stimulus from the nerve comes and stimulates the muscle, it's going to send that electrical signal along again down the length of the muscle. And what it's going to do is it's going to trigger the sarcoplasmic reticulum to release calcium ions. These calcium ions flood out into the muscle cell, and they bind to these little proteins. As they bind to those proteins, they expose the binding sites for the myosin heads. And then the myosin heads will attach, then contraction can happen. They attach, they start pulling, the actin gets closer together, contraction happens. Once that contraction is over, the stimulus is over, the calcium gets reabsorbed back into sarcoplasmic reticulum. These little proteins go back over so the myosin heads can no longer bind, and we're back to this resting stage. So in order for contraction to happen, calcium has to be released from the sarcoplasmic reticulum as well. And that's how the sliding filament theory works. So it's a conjunction between the nervous system and the skeletal muscle working together in order to cause the shortening, cause the contraction of a muscle cell.